Okay, today we're going to look at Chapter 2, the chemical context of life. So this should, for the most part, be a review for chemistry. So we'll go over some stuff, and some stuff we'll kind of skim for the most part. Okay, so when it says that biology is a multidisciplinary science, what they mean by that is that we're looking at chemistry. There are some aspects of physics when we talk about joints and anatomy and things like that. Okay, so all living organisms are subjected to the laws of physics and chemistry. So we just said. So the next slide that you're going to see use an example of formic acid by ants to maintain these devil's gardens. And these are specific for these Doria trees. So if you see here, there's only going to be one specific variety of plant. And the reason for that is that these plants have ants on them, and you would think that that would be a problem for them. But really what the ants do is they prevent any other herbivores from destroying or causing any harm to those plants. So while the ants themselves do a little bit of harm, it's minimal, and it prevents any more damage from other herbivores from coming in that area. So the ants benefit in that they get a host, they get food from the plant, but the plant benefits from not having more destruction to them. So it's kind of the lesser of two evils for the plant. Okay, so this is what they're showing you here. So they're looking at different seedlings, okay, and inside this area, if they are unprotected, what happens to them versus what happens to them when they are protected. So if you look at here, the amount of leaves that plant inside has, because it's been protected by those ants, they have far more leaves than the ones that are outside that would do that. Okay, so here's where we're going to review a lot of stuff that you should have a basis of from chemistry. So all organisms are matter. That means that we take up space and we have volume. Excuse me, we take up space and we have our mass. Okay. So matter is made with elements that you see on those periodic tables. Okay, the reason that elements are considered elements is they cannot be broken down in any way. So calcium is calcium, as opposed to something like sodium chloride or table salt. You can break that back up into its individual components. So that table salt that we just referenced, that NaCl, that would be a compound. Okay, so there's a fixed ratio of that. So when we talk about dihydrogen oxide, there are 2 hydrogens for 1 oxygen in that molecule of water. So it's a fixed ratio. Right, so a compound has characteristics that are different from its elements. So if you look here, okay, you look at that sodium and the chlorine. So individually, they have their own properties. Okay, so sodium is going to be a metal, chlorine is going to be this poisonous gas, but when you put them together, they form a very delicious treat that you can put on French fries, or better yet, tater tots, that sodium chloride. All right, so when we talk about these elements of life, okay, a lot of times you'll hear about N-CHOPs, so that would be nitrogen, and then carbon, hydrogen, oxygen, but they also usually can include their phosphorus and sulfur. So about 20-25% of 92 elements that you see on the periodic table, that is also changing as they find or make more man-made elements. Most of those are going to be the essential ones, that are 25%. So we are carbon-based organisms. We'll talk about the properties of carbon that make that so important. But for the most part, we're made up entirely of those elements. When we're talking about these trace elements, we require them, but in very, very minute quantities. All right, so here's what we're looking at those things. So you can see when you see the abbreviations, the symbols, where they get that N-CHOPs from. All right, so now when we look at these case studies, and you'll see them all throughout the book. So arsenic by itself can be toxic, but what they mean that some of these organisms are able to adapt to them, meaning that they no longer have any problems to them. The same way that you look at a relationship between a sea anemone and a clownfish. The sea anemone has these toxins, these barbs that you see on there. There's neurotoxins, but the sea anemone allows the clownfish to do it. The clownfish does not have any, or is not causing any harm by that. Okay, so we're looking at this. This is that serpentine plant community that they're referring to. So the plants in the photo are growing on that serpentine soil that you see there. Okay, this typically has things that are toxic to the plants. Okay, but you're looking at 2 things: a serpentine rock and one of the plants, the lily, and they no longer have any problem to that. So they have adapted through evolution to not be harmed by the toxins that you see there. Okay, so this concept 2.2, we talked about the atom. So the atom is the smallest unit of matter that maintains its properties. So an atom of gold would have all of the elements of gold. When you go below that, now you're looking at subatomic properties, okay, and they're not going to remain consistent there. All right, so those subatomic particles are what make up the atom. Okay, this should be a very basic review for you. So they are made up of those neutrons that you find in the middle of the nucleus. Okay, so you have your neutrons there in the middle, you have your protons in the middle, and then you have these electron clouds that are going on there. And we see that those electrons are clouds because you can't really pinpoint exactly what they are. They're constantly in movement. So you have these neutral neutrons, you have the positive charge of the protons, and then you have the electrons that are negatively charged on the outside. And this is what keeps those electrons from flying away or anything. They're attracted to that positive charge. Okay, so the neutrons and the protons form that atomic nucleus. That's where we get its mass from. Okay, and then the electrons form a cloud around there. Okay, and their mass is relatively negligible. They're so small. So when we measure those things, they're measured in Daltons. Okay, so this is what we're saying. You see that cloud of the electrons on the outside. So you have your neutral neutrons here, okay, and you have your positive protons that form the center of that nucleus. All right, so atomic number is formed by the protons. Okay, so it's the number of protons that you have. So when you see on the periodic table, that top number is usually its atomic number, and that's how they're arranged on the periodic table. When you look at its mass number, the mass number is the proton plus the number of neutrons. Okay, and then the atomic mass is approximated by that mass number. So it also includes the number of electrons that you have, or the mass of the electrons that you have there. All right, so when we talk about isotopes, if you had honors biology, we typically do in the isotope penny lab, okay, where you look at the mass of pennies that were before 1981, and then, or excuse me, if they're 1981 and older, they have a different mass than the ones that were made before that. So isotopes have the same number of protons. Okay, remember they have to have the same number of protons for them to be the same type of atom, but they have a different number of neutrons. Okay, and that becomes an issue when it starts to set off its stability. Okay, so when you look at the periodic table and you see carbon 12, what you're really talking about is that that 12 is an average of all the carbon atoms that you can find in the world. So some of them are going to be slightly less than 12, some are going to be slightly more than 12. And they'll have like carbon 14 or carbon 13. They'll have a different number when we talk about the mass there. Okay, so what do we use those things for? Those radioactive isotopes are used in dating fossils, so you can tell how old something is. Okay, you can also trace atoms through somebody's metabolic process. So you may have had an MRI or something like that that would have imaging for that stuff. And then we can also diagnose medical disorders with those. So when we're looking at them, they are looking at different cells that have used this radioactive tracer for whatever it is, okay? And it will show up differently when you run it through an MRI machine or a contrast CT or something like that, and they'll show up differently. And when they're showing these in incubators, they're showing them in different temperatures, and they can look to see when exactly. So if you're looking here, they use hydrogen. Okay, they use the radioactive isotope of hydrogen. So that will show up differently in the person's cells. Okay, so if you're looking here, this is a scintillation counter. Okay, a scintillation machine is used to detect like alpha, beta, gamma radiation. They can also be used to detect different numbers of neutrons that you have in something. Okay, so here's what you were seeing. It's just a different pictorial image of what you saw on the other slide of where DNA synthesis is most optimal. Okay. All right, so what you're looking here is a PET scan. Okay, you may have heard that before. Usually someone, if they suspect them of having cancer or they are looking to see if cancer has spread, they will ask to have a PET scan done on that individual. And a PET scan is a positron emission tomography. And basically what it does is it looks for a level of high metabolic activity in the body. And that will show up very differently as far as colors go as what you would see in a normal strip. So if you're looking here, everything else looks kind of blah, but then you can see right here that stands out really well. So that bright yellow spot that you're looking at marks an area of higher levels of radioactive glucose that they put in there. So glucose is what's used for cell respiration. So any areas that are undergoing cell respiration at a higher rate would be using more glucose. So that area would glow. So when you look back at, there's also about hotspots. They've referenced them in cancer things. All right, so when we look at these levels of energy, you would have learned this in chemistry. So energy is the ability to cause change. So potential energy is there that is stored. Okay. And then the electrons of an atom differ in their amounts of potential energy depending on where they are in that energy shell. Okay, so the state is, we said, those energy levels or electron shells. So if you look here, this would be the ball sitting up here would be a potential energy, and then it's moving through. But you're also looking at this 4 different energy levels that you have. So energy, electrons only live at fixed levels of potential energy called these shells. Okay. When you get into chemistry, they went over the different types of shells that we'll look at them, but we're not so concerned with them here in bio. So the chemical behavior is determined by the distribution of those electrons and where they are. Okay, so if you look here, this is what we're talking about before, where you have your proton numbers, okay, and you have your mass number. So remember, this is just coming from protons, and that's coming from protons plus neutrons. So when you're looking at that, here they're not showing you the mass numbers of all of them. The mass numbers would also give you information about how many neutrons you had there too. So they're showing you here all of their electron orbitals. So when things are arranged on that periodic table, they're arranged by the number of valence electrons they have because their properties are coming from that. Okay, so we just said, so the chemical behavior of an atom is determined by its valence electrons, so its properties. So whether something is a transition metal or whether it's an alkaline metal or whatever it is determined by how many valence electrons it has. And then when they talk about this chemically inert, that's your full valence shell. Okay, so those are your noble gases because they don't need anything. Their needs are all met among themselves. All right, and then we said this orbital is this 3-dimensional space where you're going to find an electron around 90% of the time. So if you were going to pinpoint it and stop it in space, you'd be able to see it there 90% of the time. Okay, so again, you would have seen these in chemistry. Okay, you do not need to memorize these now, just understand the 3-dimensionality of them. Okay, so when we look at 2-3, we're going to be referring to these chemical bonds and why they form. So anything that's not a noble gas is going to have an incomplete valence shell, which means it's going to either need to get rid of or gain electrons or share them with other atoms in order to meet their needs. So covalent, we said, is formed by sharing. Okay, this is a relatively stable bond. Okay, so when you look at these 2 hydrogens, and these are 2 nonmetals. Okay, and they come together and they're going to share the electrons in that outer orbital. All right, so that's how molecules are going to be formed. So if you're looking here, because they're only sharing one of those valence electrons, that's where your single bond comes from. But some of them are going to form more than one bond, so they can have a double bond, or they could even have triple bonds for that. So when we look at that, so we just looked at the hydrogen that you saw that, and I just drew the line in between them. So here's your hydrogen, and here's your hydrogen. That's what you're looking at here. Right? They're showing you the structural formula. But then you can also show that with the atoms themselves to form the molecule of hydrogen that you have there. Okay, now I don't think this is going to play for you, but we'll see. That may be just something that you have to have in class. Yeah, it's going to be something that you're going to have to watch in class. Okay, so when we look at these things, this is what we're just seeing on the other slide. You can see the molecular formula. This is going to show you how those electrons are divided. So you can see an electron here, you can see an electron here. Okay, you have your Lewis dot diagram, which are very easy to show because it's only hydrogen, okay, but it's showing you the valence electron on one and the valence electron on the other, and that's how that bond is formed. Okay, they're showing you the structural formula there, and then they're showing you the space filling model. So the space filling model is going to show you in proportion to one another. So if you look down at water, oxygen is a larger molecule, oxygen is a larger atom than you see of the hydrogens there. And we were talking about being carbon-based before. This is one of those things that we're showing you, that carbon can form up to 4 bonds with other atoms. Okay, so this is what we said. So compound, you have 2 or more different atoms, as opposed to just a molecule. So when you look at that CH4, that methane, or water, okay, but they just have to be different elements. And then the bonding capacity is called the atom's valence. So what would it form and why would it form that? Because if you remember for chemistry, you have this electronegativity. So you can look at the periodic table and that's going to determine how much an atom is going to attract another electron to it. All right, so if you look at this, this is that pull thing. So when we talk about either polar or non-polar covalent bonds, this has to do with how the electrons are being shared. So something is non-polar, okay, so something like an oil, they are all sharing all of the electrons equally, so there's no polarity there. When we talk about polar covalent, you're going to see a separation in charge, unequal. So if you're looking at the water, in the water molecule, the oxygen has a higher electronegativity, okay, affinity than the hydrogen do. So the electrons are going to be pulled more towards the oxygen. It does not mean that the overall molecule has a charge. It doesn't. The overall charge on that molecule is going to be zero. It's balanced. But this side has a slightly negative end, and this side has a slightly positive end overall. They're just pulled separately like that. Okay, so ion bonds are going to be different. They're not shared. They're going to be either gained or lost. So if you look at things that are on the opposite ends of that periodic table, so if you have 7 valence electrons and one has 1 valence electron, so the sodium has 1 valence electron, that chlorine has 7 valence electrons. It's a lot easier for the sodium to get rid of one of those valence electrons than it is for it to gain 7 of them. The same way it's a lot easier for the chlorine to gain 1 as opposed to losing 7. So they basically just transfer those electrons and they share, and it makes them a relatively stable molecule. However, their needs can be met by anything else. So sodium can be completed by anything else that has 7 valence electrons. So chlorine's one, but bromine would be another, bromine would be another. So anything that has 7 valence electrons would work for that. So because it's either going to donate or gain an electron, they become charged atoms because they're losing one. So sodium balance has 1 valence electron, but if it's donating one, then it becomes a +1 charge. Okay, so this is what we're just saying here. So normally, okay, it has that 1 valence electron, but if it donates that one away, it becomes positive. If that chlorine accepts it, it becomes negative. So now it's valence electron use or met, but it just changes the charges on them. All right, so this is what we were just saying. All right, so when something is positively charged, it's a cation, and something is negatively charged, it's an anion. Okay, and then the ionic bond is attraction between the two of them. So it's not as strong as a covalent bond because it can be met by several different things as long as they're in that same column on the periodic table. Okay, so again, this isn't going to play. You'll have to watch it in class. So anything that's formed by those ionic bonds, or some of the forms are called ionic bonds or salts. Okay, so you can see this brick of salt is actually formed by all the sodium and chlorine molecules bonded together. Okay, and you can see here, based on the color, that it's a 1-to-1 ratio, just like you saw there. Sometimes you'll have 2 sodiums with something else or something like that. All right, so then you have some weaker chemical bonds, and they are not as strong as covalent, they're not as strong as ionic ones, so they're more easily broken. So when we talk about these things, we're going to reference when we get to DNA and stuff like that, and they have to be weak in order for those molecules to be opened up. Okay, the covalent bonds need to be strong so that the actual nucleotides don't fall apart. Okay, so weak chemical bonds reinforce shapes of large molecules, specifically like DNA that we were just saying. Okay, and it helps the molecules adhere to each other. So those 2 strands would be an example of that. Okay, or water molecules staying together, which you'll see in the lab that we do for those. All right, so hydrogen bonds are going to form when hydrogen atoms covalently bonded to one. Electronegative atom is also attracted to another. So you'll see just like this, little charges that are connected to them. Okay, so you can see here, okay, it's not a solid line because they're not ionically or covalently bonded to it, but it's an attraction between the two of them. So this is a slightly negative end, you can see that. Okay, and this is, remember, was that slightly positive end. So there's an attraction between the two of them, but it's easily broken. Okay, and then you have these van der Waals attractions, which are they are less strong also, and they are also not as fully understood. So they can result in just, they are not hydrogen charges, but there's just, it's a weak attraction between the two of them. And one of the, I think examples that talk about that would be how a gecko's hairs stick to a wall and they can walk up them. Oftentimes what you see with geckos is that they are going up almost like a film of water. Because those water molecules are polar, the charges are unevenly distributed, and it attracts to the hairs and the geckos. Typically that would be the hydrogen traction or properties of water as opposed to van der Waals. But the van der Waals is that force that comes in that fluctuations of the charges between those molecules of the hairs, the gecko's toe hairs on the wall surface. Okay, so there it is, and its ability to climb walls or stick to water and things like that. Okay, so when we look at a molecule shape, it's usually very important to its function. So when we talk about that later on, we're going to look at proteins. And when there are issues in a protein shape, really bad things happen. So things like cystic fibrosis and stuff like that. But we said it's determined by those valence orbitals. Okay, and again, you don't need to memorize these things. We will be doing labs with them as well. Okay, so biological molecules recognize and interact with each other with a specificity based on molecular shape. Okay, meaning when we talk about cells, we have markers on our cells that identify things as self or non-self. When we're in the classroom, you can see on the cell membrane that we have there are different carbohydrate chains and there are different glycoproteins, and all those things are used for identification. And you also have molecules with similar shapes that can have similar biological effects. When we're talking about drugs, this is what we're talking about there. So drugs can mimic the shapes of other things and prevent pain or do bad things. But we're talking about the good thing here. All right, so if we look here, so natural endorphins are really good feelings. Okay, but if you look at morphine, it has a very similar structure as far as an end that would be identified by a cell. So if you're looking at these two receptors here, okay, you have natural endorphins that give you that good feeling. Okay, and then you have morphine that's also able to bind in that same thing. So morphine will affect your brain's pain perception and your emotional state because it mimics the shape of that natural endorphin that you see there. Okay, now morphine has a lot of other weird side effects that aren't mentioned here, but that's how they typically work. Okay, so when we look at these chemical reactions, okay, these are where you're making and you are breaking bonds. The big ones that we'll talk about in biology, one of the big ones will be cell respiration and photosynthesis. So you're looking at what you start with, okay, and those are going to be on the left side of your chemical equation, and then you're looking at your products that you end up with on your right side of your equation. Okay, so you're starting off with your 2 molecules of diatomic hydrogen and your 1 molecule of diatomic oxygen, and then you end up with 2 molecules of water. And if you notice, these is at a bent shape. Okay, so this is what we were just saying. Photosynthesis is an important chemical reaction. Okay, you end up starting with your carbon dioxide. You start with water. You also need some sunlight over there from the sun. That's a fabulous drawing that I got going on there. Okay, so there's your sunlight. And then you convert that to glucose and oxygen. And you're also going to make ATP from that, but it's stored within the bond, or the energy to do that is stored within the bond. Okay, so you're looking here would be another lab that we typically do. You're looking at Elodia. Elodia is a water plant, it's fresh water, and it produces sugar by rearranging those molecules that you saw from carbon dioxide and water. What you're seeing here, these little bubbles, those are air bubbles. Okay, this is going to be that sugar is going to be converted to other molecules either within the plant. All right, so all chemical reactions are reversible. Okay, so when you talk about photosynthesis, the reverse of that would be what you see happening in cellular respiration. So you're starting with glucose and oxygen, and you're breaking it down to release carbon dioxide and water, and then the ATP that we make. So chemical equilibrium is where you're seeing them in equal amounts going back and forward. Okay, so that ends where we are. And I hope that helps.